So thank you again. Our next speaker is going to be Dr. Nick Mark, who is a former graduate of our PCCM fellowship and is currently an intensivist at Swedish. And it's going to talk to us about some other information that circulates in some communities, in this case, the idea of ivermectin. So with that, take it away, Nick.
All right, well, thank you for that introduction. Can everyone hear me okay?
We can, and we can see your slides.
Excellent, okay. So since the beginning of the pandemic, 19 sessions now, this conference has been a venue to talk about evidence and therapies that have proven to be effective against COVID. But for the next 20 minutes or so, we're going to talk about a therapy that probably isn't effective. We'll break this into three parts. We'll talk about the preclinical or in vitro evidence surrounding ivermectin. We'll talk about the clinical evidence and the shortcomings of that. And then we'll conclude by talking about some of the myths and sort of cultish aspects of ivermectin in COVID. Sadly, I am not paid by the global cabal of Merck, WHO, Bill and Melinda Gates. So in order to talk about COVID therapeutics, I think it's really useful to have a framework. And this is a slide that's sort of gotten busier over the last 18 months. We can think about the stages of illness from left to right. Each represents an opportunity to intervene to alter the natural course of the disease. We can also think about therapies in terms of potential benefit. So most beneficial at the top, less beneficial towards the middle, harmful at the bottom. And we can also, as a third axis, we can think about the certainty of that evidence. So darker color, we're more certain. So as a segue to Dr. Roxby's talk, we have vaccines, highly effective in the early stages at preventing infection, preventing illness, or avoiding hospitalization. And the data behind them is very robust, so they're very dark colored. Other therapies like monoclonal antibodies against the spike protein are probably a little less effective, and the data is a little bit more up and coming. Remdesivir, which was very exciting early in the pandemic, probably has a smaller benefit. Corticosteroids have a prominent role, especially in the people who are hospitalized with hypoxia. And then there's this emerging role of immunomodulators. Tocilizumab, an IL-6 blocker, lenzilizumab, a GM-CSF blocker, and others have an emerging role in treating this disease. We also have therapies that don't work. So hydroxychloroquine most famously, but others, lopinavir, ritonavir, interferon, colchicine, and recently we've learned that convalescent plasma is probably not effective. And then just to make this more complicated, we have these sort of mixed bag therapies like therapeutic anticoagulation, which is clearly harmful in the ICU, but may confer a very small benefit in non-ICU carefully selected patients. So the question is, where does ivermectin fall on this? Is it beneficial? Is it harmful? And what is the level of evidence? Well, in order to answer that question, we have to go back to the very beginning. So in the early days of the pandemic, when that chart was completely blank, there was a frantic search going on in labs around the world for therapies against SARS-CoV-2. Many researchers use these large existing libraries of approved compounds to see if they would have any effect on SARS-CoV-2's replication in vitro. So for example, this paper by Coe et al., which was published in the Journal of Medical Virology, they took Calu-3 cells and they exposed them to different drugs at different concentrations. And then they used fluorescent antibodies against viral proteins to see if they could stop the virus from replicating. And as you see here up at the top with remdesivir, as they added more and more remdesivir to the sample, the viral proteins became less evident. And they could construct a curve that looked like this. Compare that to other drugs, lopinavir, which did affect it, but at an orders of magnitude higher concentration, or chloroquine, which was similar. So they did this for hundreds of drugs, and you can see many of them had some inhibitory effect. There are some favorites here among pulmonologists like ivacaftor. There's other ones like, among hematologists, L-trombopag. There's lots of serine protease inhibitors. Basically, there are a lot of drugs that show some inhibition of SARS-CoV-2 replication in vitro. In the context of all this work that was going on, this group at the Wagstaff lab did an experiment in viro cells. So those are monkey kidney cells. They treated them with different concentrations of different drugs and measured the amount of viral RNA using qPCR. And what they found was that the approved anti-parasite drug, ivermectin, could indeed inhibit viral replication in vitro. They calculated that the IC50, or the concentration that inhibited 50% of viral replication, was about 2.5 or 2.8 micromolar. So back in March of 2020, when this paper was first published, this was seized upon as a very exciting, promising option at a time when that table was completely blank and there were no specific therapies for COVID-19. But before you get excited about an IC50 in vitro, you always have to ask one question. What is the Cmax of that drug? So as a quick pharmacology review, Cmax is the concentration, or the maximum concentration, that's reached after administering a drug. And the idea is your IC50, which is in vitro, and your minimum effective concentration, which is in vivo, may approximate each other. And you want your Cmax to be much higher than your IC50. That way, not only does your drug reach the therapeutic level, but it's well above it. Ideally, it would be 10 times higher or even more. Unfortunately, this is not the case with ivermectin. So recall that the IC50 of ivermectin in vitro was 2.8 micromole. So it was there. But the Cmax of ivermectin is almost 2 orders of magnitude lower at 50 nanomolar. So we're using different units. It's such a big difference. In order to express this visually, we're going to have to move the axes around. So if we slide the axis way over here, we can see where the Cmax of ivermectin is. It's about 50 times too low to be effective. So this is a bad start. But it's not necessarily game over. Lots of ivermectin proponents pointed out that there is some evidence that ivermectin accumulates in the lungs, and that's just the approved dose. What if we could use a higher dose? Well, people have looked at that as well, using data from cows where they can actually measure the tissue-specific concentration of the drug. They calculated that the Cmax in lung tissue is 80 nanomolar instead of 50 nanomolar. So it's higher, but not a game changer. They also explored, what if you used 10 times the usual dose of ivermectin, 120 milligrams instead of 12? Well, this gets you up to a Cmax of 0.8. But at this point, you're kind of at risk of causing toxicity, and you're still well short of where you need to be. So in conclusion, even though there was a promising signal in those monkey kidney cells, the pharmacokinetics just don't look great. Even if we make really generous assumptions about how it's going to go in the different tissues, and even if we use higher than usual, higher than approved doses, we're still not quite getting there. Despite this, this became the highest impact paper ever published in antiviral research. You can see not only was it highly cited in just a year, but it was highly shared on social media. Many authors were quick to question these things, though, and they wrote some very good letters to the editor about this. And to their credit, the authors of that paper, the Cayley et al. Wagstaff paper, really acknowledged these criticisms. And I'll just read a couple of bits here. Yao and Knoll, who wrote those letters to the editor, aptly point out that the published studies show that blood levels of ivermectin achieved during standard therapy are much lower than the concentrations we reported as inhibitory. They go on to say that if concentrations of ivermectin that were high enough could be achieved, they would likely be toxic. And they say, these points are well made and we are in agreement. They also point out that because ivermectin is actually inhibiting a cellular target, not a viral target, toxicity is likely, especially at higher doses. They conclude by saying, finally, it is critically important to remember that ivermectin as an antiviral is in a very early phase. Under no circumstances should anyone self-medicate with the drug. To their credit, on Dr. Wagstaff's website and on every press release that her lab puts out about this, they always start with this warning: do not self-medicate. And they always inject caution into what they say, saying things like, ivermectin is very widely used and seen as a safe drug. We need to figure out now whether the dosage you can use in humans will be effective. That's the next step. So she's not making any big claims here. Unfortunately, there are other people who are. And this kind of brings us to the clinical trials of ivermectin, which is where the train really starts to jump the tracks. So proponents of ivermectin will tell you that there have been dozens of trials, and that is correct. However, very few of these are methodologically rigorous. So if you want to go over these in detail, I recommend this resource here. The NIH COVID treatment guidelines have a great description of each of these trials, and you can look it up and you can see. Just to summarize, though, some of the criticisms that kind of limit the applicability of these studies. So most of these studies are preprints. Very few of them have been peer reviewed. And that was expected a year ago, but the fact that some of these papers are languishing without being published for over a year suggests that there are major concerns that peer reviewers are uncomfortable with. Second, most of these studies are retrospective and observational. None of the observational studies was pre-registered as far as I can tell, so there may be significant publication bias. And furthermore, many of these studies are really small, just like a couple dozen patients, 24 in one case. And so their power is really questionable as well. Third, they varied significantly in intervention. Some of them used ivermectin as prophylaxis, others used it as treatment. Some used standard doses, some used really high doses. So with that kind of heterogeneity, it's really hard to put these studies together into a cohesive picture. Another issue is the endpoints. So most of these studies used non-patient-centered endpoints, like viral clearance by PCR as the primary endpoint. There were a few that used other criteria like, quote, clinical recovery, but the paper that used that didn't actually define it. So we don't know what that means, which makes it hard to interpret. Finally, there are some really significant confounders in these putative positive studies of ivermectin. In some cases, the control groups were things like hydroxychloroquine, azithromycin, doxycycline. So things that we know don't work were the control. So that could bias towards seeing an effect. And in other cases, there was unequal use of other treatments. So for example, in a couple studies, the ivermectin arm had greater use of corticosteroids, which we know is one of the most effective therapies to date in this disease. The bottom line is that although there are numerically many studies of ivermectin, there are very few that add much certainty to this conversation. Most of them have major methodologic flaws and they're hard to interpret. There is one study which I think is the exception to this that I want to highlight. This is a study called the EPIC trial. It was looking at the effect of ivermectin on time to resolution of symptoms among adults with mild COVID. This was a double-blind RCT done in Cali, Colombia. They identified outpatients using a government database who had recently tested positive, reached out to them to enroll them in the study, and they received either ivermectin, 0.3 mg per kg for 5 days, or placebo. The primary outcome was time to symptom recovery. So they did this study, they randomized 400 patients, and they found absolutely no difference in time to symptom recovery. As a secondary endpoint, they looked at clinical deterioration, visits to the emergency department, escalation of care like being hospitalized, and mortality. And there was no difference in any of those secondary endpoints either. Importantly, they didn't notice any increase in adverse effects or serious adverse events in the ivermectin group, suggesting that at least in this relatively well population receiving a standard dose of ivermectin, there were no significant toxicities. So in summary, this is one of the few really good papers that we have that's actually been peer reviewed, it's actually randomized, it actually used a good placebo group, and it actually had enough N to really tell us something. And this was a profoundly negative study of ivermectin. So while this doesn't foreclose the possibility of some clinical benefit, it probably puts an upper limit on the effect size. No matter what studies are done in the future, I'd be shocked if there was a big effect size having seen this study. Fortunately, though, this will not be the last good study. Even though there have been something like 60 low-quality studies published, there are many good ones in the works. There's currently 71 trials of ivermectin and COVID registered on clinicaltrials.gov. And we just talked about EPIC, which was just published a few weeks ago, but there are many other good ones. So for example, the ACTIV-6 study, which is comparing ivermectin versus placebo in 15,000 participants in the US with mild to moderate COVID, will probably give us a lot of information. In this study, patients who test positive for COVID will have placebo or study drug mailed to them. And there are many more well-designed placebo-controlled studies going on in South America. Ivercol in Colombia, Ivernex and Ivercor COVID in Argentina. Ivercor COVID is looking specifically at whether ivermectin can prevent hospitalization and whether it can reduce mortality. And then in Brazil, there's a large study, which unfortunately does not have a catchy acronym, of repurposed approved therapies. That's looking at not just ivermectin, but also fluvoxamine, metformin, and a placebo. So with these high quality studies currently recruiting, I think it's likely that we will have a lot more good data in the coming months. Until then, however, I don't think meta-analysis is going to be terribly fruitful. And that brings us to our next point. So if you've spent much time on the internet reading about ivermectin, it's likely that you've come across some of these dubious meta-analyses. For example, this one here claims that when you put all these low quality studies together, there's a 96% reduction in mortality. They do acknowledge that there's a chance that they could be wrong about this and estimate that it is 1 in 2 trillion. So I'm not going to dunk on this paper too much more, except to say that every time somebody reads this sentence, Thomas Bayes probably rolls over in his grave. I think this also highlights why one of the first steps in any legitimate meta-analysis is to do a risk of bias assessment and make sure that you're only including high quality studies. Because when you include studies that don't have control groups and have significant confounders, you get results like this, which are frankly uninterpretable. There's a great article in the BMJ's evidence-based medicine subjournal, which makes the point that, quote, unpublished colorful forest plots on these websites have many problems. They are not pre-registered, so they can change their methods willy-nilly. They don't define inclusion-exclusion criteria, and they don't have rigorous statistical means to address uncertainty, as we saw on that last slide. But I think what really worries me about this is that by performing these, by sort of providing the veneer of rigor, these ersatz meta-analyses confuse people. And this article points out that that's confused clinicians, patients, and even policymakers. People get this emailed to them and it superficially looks like it's well done. And you have to spend a little bit of time to really see the flaws. When you do, it's very clear. This is an example of garbage in, garbage out. If you meta-analyze bad studies, you get meaningless results. But I think these in particular have fooled a lot of people. Now that we've talked about the clinical data, let's conclude by talking about some of the kind of worrisome cultish aspects of this. So the group that has advocated for ivermectin, the Frontline Critical Care Clinicians in COVID, or FLCCC, was founded by Dr. Paul Marik. You may remember Dr. Marik from 2017 when he claimed to have cured sepsis using a cocktail of vitamin C, thiamine, and hydrocortisone. He went on in 2018 in a press interview to say that not one person had died of sepsis at his institution since beginning use of what he called the metabolic cure for sepsis. Despite Dr. Marik's enthusiasm, 8 large RCTs have failed to replicate his findings, and the consensus is that there is no benefit to vitamin C or the metabolic cocktail in sepsis. The co-director of FLCCC, Dr. Pierre Corey, testified before Congress, the Homeland Security Committee, and described ivermectin as, quote, a miracle drug. He has categorically dismissed any negative studies of ivermectin and has derided organizations like the WHO and the NIH as being compromised by pharma or this larger cabal. What's really worrisome to me about this is that it's unclear what, if anything, that could be published that would dissuade these people from their belief that ivermectin cures COVID. Finally, ivermectin proponents have significant overlap with other fringe medical beliefs. A lot of people who believe in ivermectin also believe in hydroxychloroquine. Many are anti-vaxxers and many are conspiracy theorists. Now, while this in and of itself is not a bad thing, right? I mean, flat earthers can get stuff right too, I suppose. The issue is that this ivermectin is being used and embraced by anti-vaxxers as a means to discourage vaccination. Many proponents of ivermectin are very careful not to be too critical of anti-vaxxers because they're their supporters. And what this leads to is a situation where whether ivermectin is effective or not, the resistance to vaccination that accompanies it is probably harmful. And I worry that in the coming months, as we see more spread of like the infectious Delta variant, and we see vaccine hesitancy because of ivermectin, that ivermectin, whether it is effective or not, is going to cause real harm. So to summarize what we started off in vitro, and we talked about how there's this large discrepancy between the dose of ivermectin that is effective in cell culture and what is actually achievable in plasma, and how they're widely discrepant. Next, we talked about the clinical studies and how there's really very little we can take away from most of them, but there are high quality studies in the works. We talked about some of the other aspects of this movement that are a little bit alarming. And I'll leave you with the guidelines here, which are basically that no organization recommends the use of ivermectin. 2, the WHO and the IDSA specifically recommend against, except in the context of a clinical trial. The FDA has issued a warning about its use, particularly the veterinary forms of it. So I was going to end there, but I want to just share one quick thing, which was in the last 24 hours, noted world-renowned evidence-based medicine expert Joe Rogan has decided to weigh in on ivermectin. His podcast here has 16 million subscribers. So this is reaching a larger and larger audience every day. I hope that I've given you some facts so that you'll be able to respond to questions by patients, family members, and perhaps some of our peers who have heard about ivermectin from Joe. Thank you very much for your attention. I'm really excited to talk to you and answer your questions. You can download this talk if you go to this QR code, or if you go to my website. If you want to play with these slides yourself, you're welcome to.
Thank you so much, Dr. Mark, for giving us some of the tools to help combat some of the nonsense around some of the drug information out there now, and for your work, in the Twitterverse and elsewhere today to help with cutting through that. I will ask you